So if you haven't looked at the first video, which is the investment phase of glycolysis, go ahead and check that out. All right, but if you have already watched that video, then stay tuned. Let's dive right in. In order to move forward with glycolysis, we had to use 2 ATP in order to move forward with glycolysis. So in this phase, get the payout for that. We're going to get paid our ATP back and then some. You also remember from the investment phase, the dihydroxyacetone phosphate was converted into glyceraldehyde 3-phosphate by the triose phosphate isomerase. I also, shorten it just that isomerase. So now we have 2 glyceraldehyde 3-phosphates. That's why you see the 2 right there. So just know as we move through these reactions, there are 2 of each. So now we have glyceraldehyde 3-phosphate, and it's going to be processed by glyceraldehyde 3-phosphate dehydrogenase. We're going to be losing hydrogens. 2 NAD molecules are then converted into 2 NADH. And where did the H come from? From the dehydrogenase. So let's move on to step number 7, where we have 1,3-bisphosphate glycerate. And in the bond, the enzyme phosphofructokinase, the enzyme that helps transform this into 3-phosphoglycerate. So that is the molecule that helps transform 1,3-bisphosphate glycerate into 3-phosphate glycerate. It takes away a phosphate from the bisphosphate glycerate or phosphate groups on this one glycerate. That's why you have the 1 and the 3. Moves it to ADP, which transforms it to ATP. Let me back up, make sure those arrows don't confuse you. But yeah, so this the phosphate group on the first carbon is stripped away or moved from the bisphosphoglycerate and it's moved on to the ATP. And after that, you will have 3-phosphoglycerate. That means there's a phosphate group, the third carbon. In order to transform 3-phosphoglycerate into 2-phosphoglycerate, we're going to use mutase. We're going to shift that phosphate group from the third carbon to the second carbon. Mutase basically means to shift, move it around. The phosphoglycerate mutase does, it just transfers the phosphate group from the third carbon to the second carbon on that same phosphoglycerate molecule. We're almost done. We're almost to our pyruvate. But in order to get there, we have to use this enzyme called enolase. The enolase is going to dehydrate the 2-phosphoglycerate, and so we're going to produce a water molecule from this. There's 2 because you still have 2. So now that we have enolase, or now that enolase is dehydrated our 2-phosphoglycerate, that will now transform it into phosphoenolpyruvate. We're almost done. This is our 10th enzyme, pyruvate kinase, from 2-adenosine diphosphate and transformed into 2-adenosine triphosphate. So we have removed our phosphate group and put it on ADP to make it ATP. We will now have our final product, pyruvate. That is the overall process of glycolysis. The reactions of glycolysis produce a net yield of 2 ATP molecules and 2 NADH molecules for each molecule of glucose. So our net total, so say for instance, it took you 2 ATP to produce the first half of glycolysis, and you end up getting back 4 ATP from the latter half of glycolysis, well, then your net ATP would be 2, because 4 minus 2 equals 2. So even though you got back 4 ATP, you had to use 2 ATP in order to get to this position. So something to keep in mind. It's the same thing as business. Like, say for instance, you made a product and it cost you $2 to make, and then you sold it for $4, right? So someone paid you $4. So yay, great, you had $4, but you spent $2 to make the product. So not only did you have to, get that money back that you used during your investment, you are now left with $2. So That's the best way that I can describe it. That's the basic thing here. So you have 2 ATP, and you also gained 2 NADH. So that's like a little bonus, like, hey, I didn't pay for your product, but I wanted to donate to your business. Oh, well, thank you. I'll take that too. That's what this 2 NADH is like. All right, so these are some questions I wanted to ask at the end of the video. Pulling from Glycolysis Part 1, the investment phase. Why is glycolysis important? What are the 2 molecules produced by aldolase? So for glycolysis, part 2, the payoff phase, how many ATP are produced in the payoff phase? Don't get this confused with the ATP you invested from the investment phase. If you're talking about the net amount of ATP, then yes, it would be 2. All right, number 2, this enzyme shifts a phosphate group of 3-phosphoglycerate to 2-phosphoglycerate. Which enzyme is this? All right, and for the third and final question, NAD plus is reduced to, meaning that hydrogens are added, NAD plus is reduced to what? Guys, that's it for the medical questions. Now here I just want to give you some medical application. I want to talk about fluoride in toothpaste. It helps strengthen dentin and enamel inside your teeth, but it also helps reduce the enolase, and it also inhibits bacteria from forming, you guessed it, cavities. So next, you have phosphofructokinase 1 deficiency. So that's the most highly regulated enzyme in glycolysis. If this is deficient, that means you'll have an inability to store glycogen, which is awful because this leads to exertional fatigue and muscular exercise intolerance. All right, and for the final one, pyruvate kinase deficiency. If this is deficient, your red blood cells will break down much faster than normal, and that can pose an issue in receiving the amount of oxygen you'll need to survive. If you like this video, please give it a thumbs up. If you learned something from it, please share with a friend, and please subscribe to the channel and hit the notification button so you don't miss an upload whenever I upload a new video.